Hey, hello again. I have a question. Do you know what chromosomes are? What are they for? That their number varies from species to species. That their number has changed in humans through evolution. Well, let's discover together some amazing things about that small invisible world. First, let's see what chromosomes are and what role they play. Chromosomes are macromolecules, corpuscles, that are located inside the nucleus of a cell and represent the carriers of hereditary genetic traits. They become visible under the microscope during cell division, when the chromosomal material, chromatin, spiralizes and packages. Chromosomes are made primarily of deoxyribonucleic acid, DNA, the molecule that carries the genetic code, or genes, protein, histone and non-histone. Proteins help package and organize the long strand of DNA into a compact chromosome structure. Each chromosome before cell division consists of two identical longitudinal halves, called chromatids, which are joined at a place called the centromere. The main role of chromosomes is to ensure the precise transfer of genetic material from the parent cell to the offspring cells during cell division, mitosis and meiosis. The number and shape of chromosomes are specific for each type of organism. Human somatic, body cells contain 46 chromosomes, organized into 23 pairs. 22 pairs of autosomes, body chromosomes, which are common to both sexes and are numbered from 1 to 22. One pair of sex chromosomes, gonosomes, which determine sex. Women have a pair of XX chromosomes. Males have a pair of XY chromosomes. And now, let's go back a little further and look at what happened through evolution. Through evolution, chromosomes underwent significant changes in number, structure, and organization, which was a key factor in the emergence of new species, speciation, and the diversity of life on Earth. Major processes and changes include changes in the number of chromosomes, numerical aberrations, polyploidy. Duplication of entire sets of chromosomes is a frequent occurrence in the evolution of plants, where the mechanism of speciation is extremely important and less common in animals. Aneuploidy, changes in the number of individual chromosomes, missing or extra chromosomes, are generally harmful, but rarely may have played a role in evolutionary change. Fusion and fission, joining two chromosomes into one, fusion, or splitting one chromosome into two, fission, are changes that change the total number of chromosomes in a species karyotype. The most famous example in humans is the Robertsonian translocation, which has resulted in humans having 46 chromosomes, while our closest relatives, chimpanzees, gorillas, and orangutans, have 48. Deletions and duplications. Loss or duplication of parts of a chromosome. Duplications of genes and entire chromosomal segments are extremely important for evolution because they create new genetic material that can evolve into new functions without damaging the original gene. Inversions, turning a chromosome segment 180 degrees. These changes can affect the crossing over of chromosomes during meiosis, which can lead to reproductive isolation and the formation of new species. Translocations, exchange of parts between non-homologous chromosomes. All these changes, mutations, are the source of genetic variability, which is affected by natural selection and other evolutionary factors, thus shaping the genetic material of species over millions of years. Let's stay here a little longer. Let's see what exactly the process of chromosome fusion represents. A process of chromosome fusion that occurred in human evolution led to the formation of human chromosome 2, a key event that explains why humans have 46 chromosomes, 23 pairs, while our closest relatives, such as chimpanzees, gorillas, and orangutans, have 48 chromosomes, 24 pairs. The fusion is thought to have occurred when two smaller chromosomes, which existed in our common ancestor with other great apes, and which still exist today as separate ones in chimpanzees, e.g. chromosomes 2A and 2B, joined end to end. This event was a unique mutation that occurred in a single individual within a population of early hominins, about 0.74 to 4.5 million years ago. In order for this change to become fixed in the population, an individual with one fused chromosome, heterozygote, had to reproduce. And over time, individuals with a pair of fused chromosomes, homozygotes, had to appear, which ultimately led to the establishment of a new number of chromosomes in the entire, later reproductively isolated population. So, do we have evidence for that? The most compelling evidence for this event comes from a direct comparison of the human genome with that of the great apes. Matching chromosome bands. When the staining patterns, bands, of human chromosome 2 are compared, they match perfectly with the patterns of 2 separate chromosomes in the chimpanzee and other great apes. Presence of a rudimentary centromere. Chromosomes usually have a single centromere, site of chromatid attachment, crucial for chromosome movement during cell division. Human chromosome 2 has one active centromere, but also contains the remains of another, inactive, rudimentary or fossil centromere in the place where the centromere of one of the original chromosomes was. Telomere remnants in the middle. Telomeres are the protective ends of chromosomes, composed of specific repetitive DNA sequences, TTA-GGG. Under normal conditions, telomeres are found only at the ends of chromosomes. However, in the middle of human chromosome 2, at the point of fusion, remnants of telomeric sequences were found, and in the reverse orientation, as expected in end-to-end fusion. Finding these internal telomeric sequences is a strong genetic fingerprint of an ancient splicing event. This evidence provides a plausible explanation for the difference in chromosome number and is considered one of the strongest lines of evidence for the common ancestry of humans and great apes. This event was a critical step in human evolution, potentially affecting reproductive isolation from other populations and contributing to the further development of distinctive human traits. Now let's look at something really interesting. Let's look and compare the number of chromosomes of humans with some types of animals and plants. Wait a minute. As far as I can see, man and potato have a similar number of chromosomes, while a dog has much more. And yet the difference is so significant? How come? The number of chromosomes in different species, despite the fact that it may seem arbitrary, does not determine the complexity of the organism. This is a common myth in biology. As you may have noticed, a potato has more chromosomes than a human, and a dog significantly more than both. However, the complexity of a living being does not depend on the number of packages, chromosomes, but on the content of those packages. That is, on the total amount of DNA and the number of genes, as well as on the way in which these genes are regulated and expressed. The complexity of the organism primarily results from gene counts. Although potatoes have a similar number of chromosomes, humans have a significantly higher total number of genes. Estimates range from 20,000 to 25,000 for both humans and potatoes, but with much greater diversity in the human genome. Gene regulation. The biggest difference lies in how genes are turned on and turned off. Gene expression. The human genome has much more complex regulatory mechanisms that enable the development of complex structures, such as the brain, nervous system, and various tissues and organs. Amounts of non-coding DNA. Large parts of the genome, especially in mammals, consist of DNA that does not carry instructions for proteins, but has important regulatory functions or other roles that contribute to complexity. In short, the number of chromosomes is just the way the genetic material is physically packaged. It doesn't matter how many packages the encyclopedia is packed in, but what it says. Human complexity arises from the content of genetic information and the complexity of its regulation, not from the number of chromosomes itself. And that's all for now. Thanks for watching, and see you again soon!